Welcome to another important topic regarding this blood group, that is Bombay blood group. Very interesting and important topic. This is the story of antigen H, small h. Normal people who are called as a non-Bombay, their genotype is capital H homozygous dominant or heterozygous dominant. These are normal. The antigen present in their body is capital H. This capital H antigen is very important in the body because this synthesis of this antigen H is located on the chromosome number 19. Those alleles, two alleles are expressed either capital H or capital H or small h. However, this H antigen is synthesized in the blood. This is located on the red blood cell. Bombay blood group is not a separate blood group. He may belongs to A group, B group, AB group or O group. Besides these AB antigens, another antigen is also present on the red blood cell, that is the H antigen, which is determined by the genotypes or alleles, homozygous dominant or heterozygous dominant. What is the importance of H antigen is, if the H antigen is present on the red blood cell, that can promote the synthesis of AB antigens. For example, if he belongs to A group, if he is containing H antigen on his red blood cell, this H antigen can stimulate the synthesis of A antigen. If there is no H antigen, the person could not synthesize A antigen, as well as on the B also. If the H is present, B antigen is prepared. If the H is absent, the B antigen is also not prepared. In the AB group person also, H antigen has a lot of importance in synthesizing the AB antigens also. Absence of H antigen could not promote the development of other antigens also, AB antigens. That is the importance of H antigen. In the Bombay group people, they may belong to any group, A, B, AB or O group. If their genotype is small h and small h, mutant, they could not synthesize the H antigen. There is no H antigen on the red blood cell. If there is no H antigen on the red blood cell, they cannot synthesize AB antigens also. They may belong to A, B, AB and O group. Actually, they have to belong to any one group of these four, but they could not synthesize any A and B antigens on their red blood cell. Then in the clinical test, they would be identified as the O negative person, but he is also not belongs to O negative. That is the main discussion. How this Bombay genotype is forming, I will discuss. A patient was joined in the hospital. The same group blood is transfused into the patient body, but the patient felt into risk. Then they started research on the blood groups. Finally, they found this H antigen. How many Bombay blood group persons are there throughout the world? India, 0.004, means only 4 million population are the Bombay blood group in the total human population throughout the world. But whereas in the India, 1 in 10,000. In the Europe, 1 in 1 million people are becoming as Bombay blood group. This Bombay blood group is first identified in the Bombay by the scientist Bende in a particular community of the people, those who are following the consanguineous marriages. So in the Mumbai, the percentage is very high than world population, that is 0.01. This is important. So the Bombay blood group is first identified in Mumbai, India, and the most of the persons who are having with the Bombay blood group is, most of them are living in the Mumbai. But this Bombay blood group could not cause any disease. They are normal persons. But when they require the blood group, they have to take it from the Bombay blood group only. They have to donate the blood to Bombay group persons only. That is the importance of Bombay blood group. Then we will discuss about them in detail. Here are some principles regarding this discussion. Bombay blood group. Bombay blood group doesn't have H antigen, capital H antigen on their RBC, but they are having anti-H antibodies in their serum. The B lymphocytes can synthesize this anti-H antibody in the plasma because H antigen is absent in that body. So this H antigen is precursor of the AB antigens allelic expression. Means the red blood cell of the normal person, a non-Bombay person, should have capital H antigen on his red blood cell. Then he can synthesize the AB antigens on the red blood cell. If the H antigen is absent, the person could not synthesize AB antigens also. This means the H antigen is the precursor of AB antigen. Alleles are expressed at the presence of this H antigen only. So the Bombay blood group genotype is small h. This is mutant type. Bombay blood transfusion. Who can give the blood to the Bombay? Bombay can give the blood to Bombay only. Bombay can receive the blood from Bombay only. That is the importance of blood transfusion belongs to this Bombay blood group. Then what is the genetic makeup of H genes or non-Bombay and Bombay genes? Genotypes. On the chromosome number 19, H antigen synthesizing alleles are located. In the normal people, homozygous dominant, the same loci, two dominant genes or alleles are located. Those are capital H and capital H. These can synthesize the capital H antigen. In some people, the homologous pair of chromosomes are containing capital H and small h. These are the heterozygous population, but H is the dominant, so he can also synthesize the capital H antigen on his red blood cell. These two are non-Bombay or normal persons. But in the Bombay blood group, on the homologous pair of chromosomes in 19th chromosome, the two alleles are mutant alleles or double recessive alleles. These double recessive alleles could not synthesize H antigen on the red blood cell, so the red blood cell does not contain any H antigen. He cannot synthesize any AB antigens also, but he can possess some anti-H antibody in his body. So the homozygous dominant is non-Bombay, capital H, capital H. Heterozygous dominant is non-Bombay, but one is dominant allele, another one is a recessive allele. But homozygous recessive, this is called double recessive, mutant one, small h located on the homologous pair of chromosomes. These are Bombay blood groups. In the non-Bombay candidate, the normal individual, the H antigen is prepared in the body that is responsible for the complete synthesis or development of AB blood groups. Means absence of H antigen could not promote the development of AB antigens on the red blood cell, so the AB antigens are not synthesized. No blood antigens are found on the RBCs. Then what is their group in clinical test? That is looking like a O negative person, but they are not belongs to the O negative also. Inheritance pattern of the Bombay blood groups. This is an important example discussed. The mother and father both belongs to AB group. On a clinical test, the child's group was identified as neither A group nor B group or AB group. The child group is not identified as A group, B group or AB group. Instead, the clinical test is showing as O group. Is it possible? Analyze the inheritance pattern. First we will see the mother and father. What will be the blood group pattern of the child? Mother, father, AB group and AB group. The child should have AA, AB, AB or BB. There is no chance for the O group. This is our normal calculation. So the mother's blood group is AB, father's blood group is also AB. But these are non-Bombay candidates, normal people. In the mother and father, the H antigen is also present. This H antigen genotype is heterozygous. So what type of alleles are going to be formed at the time of meiotic division? Capital A, capital H gamete, capital A, small h gamete, capital B, capital H gamete, and the capital B, small h gamete are produced in the mother. In the father also same type of gametes are formed. Those are the sperm cell. 4 types of ova, 4 types of sperm cells are synthesized. Now we will arrange these 4 types of sperm cells and egg cells are their genotypes, haploid genotypes on the checkerboard or Punnett checkerboard. Here the sperm cells are arranged, here the egg cells are arranged. Then you can start writing the genotypes. AH, this one, AH, capital A, small h, this one. Likewise, you can write all the genotypes, 16 genotypes. In these 16 genotypes, the 6th one, 8th, and the 14th, 16th are showing in the red color because they are having mutant double recessive H alleles, small h alleles. What are they? 6th, 8th, 14th and 16th. What was the genotype? Capital AA, HH. 8th one is ABHH. 14th one is also ABHH. 16th one is BBHH. Right? This 6th person has to belongs to A group because 2 dominant A alleles are there, but on his red blood cell do not contain the H antigen double recessive condition. So A antigen is also not synthesized. So the red blood cell is looking like this. He belongs to 8th person AB group, but the red blood cell is showing buried like this. AB antigen is also not synthesized due to the absence of capital H antigen. In the 14th person is also belongs to AB group, looking like the 8th group person, and the 16th one has to belongs to B group, but the red blood cell does not contain any H antigen, so the B antigen is also not synthesized. The red blood cells are showing like that. So in the clinical test, they were determined as the O negative persons, but they are also not belongs to O negative. They are special Bombay group. Means, for example, if you will give the O negative blood to the Bombay person, what will happen? The risk is generated in the Bombay blood group person recipient, because the Bombay blood group person may not have any antigen on his red blood cell, but he has anti-H antibody in his plasma. So the O negative person may not have A antigen, B antigen or H antigen, but he is having H antigen because he is a non-Bombay candidate. H antigen is present. If this blood enters into this Bombay blood group person, anti-H antibody agglutinate with the H antigen, the blood in the small blood vessel, the blood is clotted and clumped, the patient can be entered into the risky. So the Bombay blood group person is unique or special, not belongs to O negative also. Final question is, who can give the blood to Bombay person? The answer is Bombay can donate the blood to Bombay. Bombay can receive the blood from Bombay only. That's it. Within 2 minutes, I'll try to recapitulate what we have learnt is Bombay blood group persons has a small h double recessive condition, but the normal non-Bombay candidates are homozygous dominant or the heterozygous dominant. Most of the world population are non-Bombay. Only few of them, very less percent are belongs to Bombay blood group. Bombay blood group is identified in the Mumbai in the 1952. Bombay blood group person do not have H antigen on their red blood cell, but they are having anti-H antibody on their plasma. H antigen is must and should present on the red blood cell to synthesize the other antigens A and B. Bombay genotype is small h. Bombay can donate the blood to Bombay, and Bombay can receive the blood from Bombay only.